Have you ever wondered how methamphetamine affects the brain? If you have, then you're in the right place. Let's delve into this potent psychostimulant drug and its profound effects on our brain's inner workings. Methamphetamine, or meth as it's often called, is no ordinary substance. It's known for its ability to wreak long-lasting havoc on the brain, with the dopaminergic and serotonergic systems bearing the brunt of the damage. These systems are instrumental in regulating our mood, memory, pleasure, and reward mechanisms, among other functions. When they're disrupted, the consequences can be dire. But how exactly does methamphetamine inflict this damage? Well, the drug stimulates a massive release of dopamine, the neurotransmitter responsible for feelings of pleasure and reward. This dopamine tsunami doesn't come without a price though. The excess dopamine undergoes autooxidation, leading to the formation of harmful compounds known as reactive oxygen species and quinones. These substances are pretty much unwelcome guests in our brain, causing damage to the very terminals that release the dopamine in the first place. Beyond the dopamine surge, methamphetamine also triggers an excessive release of glutamate, another neurotransmitter. This overstimulation of glutamate receptors can lead to excitotoxicity, a fancy term for a process that can result in neuronal death. This is akin to a partygoer who overstays their welcome and ends up causing harm to the hosts. Methamphetamine doesn't stop there. It also impairs mitochondrial function, the powerhouse of our cells. Plus, it activates microglia, the resident immune cells of the brain, which release pro-inflammatory substances, exacerbating the neuronal injury. Add to this the structural changes that methamphetamine abuse can cause, including reductions in gray matter volume and white matter integrity in various brain regions, and the picture becomes clear. Methamphetamine is a force to be reckoned with, causing a myriad of changes that can lead to cognitive impairments and other neuropsychiatric consequences. These effects set the stage for the neurotoxic impacts that methamphetamine can have on our brain. This is the tip of the iceberg when it comes to understanding methamphetamine's effects. But as we peel back the layers, we begin to see the full extent of this drug's devastating impact. Methamphetamine causes a massive release of dopamine from presynaptic terminals. And here's where things begin to unravel. This excess dopamine doesn't just sit idle. It undergoes a process called autooxidation. Now, this isn't your everyday oxidation. It's a biochemical reaction that leads to the formation of highly reactive molecules known as reactive oxygen species, or ROS, and quinones. These oxidative species are akin to molecular wrecking balls. They wreak havoc on the dopaminergic terminals, the very nerve endings that release dopamine. This damage to the dopaminergic terminals can lead to neuronal degeneration, essentially causing the nerve cells to deteriorate and ultimately die off. So, it's not just the overabundance of dopamine that's problematic, it's the chain reaction it triggers, culminating in oxidative stress and neuronal damage. This intricate yet destructive process underscores the role of dopamine and oxidative stress in the neurotoxicity of methamphetamine. This is how dopamine and oxidative stress play a role in methamphetamine's neurotoxicity. Methamphetamine exposure also results in excessive glutamate release. Now let's think about this. Glutamate is a neurotransmitter that plays a key role in normal brain function. But when there's too much of it, it can overstimulate the glutamate receptors. This overstimulation, or excitotoxicity, triggers a cascade of events. It causes a surge in calcium influx, leading to mitochondrial dysfunction and ultimately neuronal death. Now let's delve a little deeper into mitochondrial dysfunction. Mitochondria are the powerhouses of our cells, responsible for energy production. Methamphetamine, however, throws a wrench in the works. It impairs mitochondrial function by inhibiting complex 2 of the electron transport chain. This disruption in energy production, coupled with the increased oxidative stress we discussed earlier, contributes to neuronal damage. So, in essence, methamphetamine's harmful effects on the brain aren't just limited to dopamine release. It's a complex interplay of excitotoxicity and mitochondrial dysfunction. This is how excitotoxicity and mitochondrial dysfunction contribute to the neurotoxic effects of methamphetamine. Methamphetamine induces the activation of microglia, the resident immune cells of the brain. Now let's delve into the intriguing world of neuroinflammation, where the brain's defenders can become its assailants. Microglia, usually the brain's guardians, spring into action when methamphetamine enters the scene. These cells, akin to the brain's own police force, become activated, shifting from their typical, peaceful state into a more combative form. However, just as a city in constant high alert can suffer from the stress, so too can the brain. In their activated state, microglia release pro-inflammatory cytokines, a kind of distress signal, along with other neurotoxic factors. Picture this like a police siren blaring non-stop, creating an environment of sustained stress. This constant state of emergency exacerbates neuronal injury, contributing to the neurotoxic damage caused by methamphetamine. But the impact of methamphetamine doesn't stop at neuroinflammation. This powerful psychostimulant also instigates structural changes within the brain. Imagine for a moment the brain as a bustling cityscape, with gray matter as the buildings and white matter as the roadways connecting them. Chronic methamphetamine abuse can lead to a decrease in the volume of these buildings, the gray matter, and a loss of integrity in the roadways, the white matter. These changes can occur across various regions of the brain, akin to multiple districts of a city facing simultaneous decline. Such structural changes may be behind the cognitive impairments often seen in chronic methamphetamine users. Much like a city with crumbling buildings and potholed roads struggles to function efficiently, these brain changes can lead to deficits in memory, decision-making, and executive functions. To conclude, methamphetamine's influence on the brain extends far beyond its initial neurochemical effects. It triggers a cascade of events, from neuroinflammation to structural changes, which collectively contribute to its neurotoxicity. This is how neuroinflammation and structural changes contribute to the neurotoxicity of methamphetamine. As we have seen, methamphetamine's neurotoxicity is a complex process involving multiple mechanisms. Beginning with a surge in dopamine release, the drug triggers oxidative stress through autooxidation, leading to the formation of damaging reactive oxygen species and quinones. This is followed by excitotoxicity, where excessive glutamate release overstimulates receptors, causing calcium influx, mitochondrial dysfunction, and eventually neuronal death. Further, methamphetamine disrupts mitochondrial function, impairing energy production and increasing oxidative stress, which further contributes to neuronal damage. Simultaneously, it activates microglia, the brain's resident immune cells, causing the release of pro-inflammatory cytokines and other neurotoxic factors that exacerbate neuronal injury. Finally, chronic use of methamphetamine leads to structural changes in the brain, reducing gray matter volume and white matter integrity. The extent of neurotoxicity varies depending on factors like dose, frequency of use, administration route, and individual genetic and environmental factors. While more research is needed, understanding these mechanisms can help in developing effective treatments for methamphetamine addiction and its associated neurotoxic effects.